Hello and welcome everyone to my YouTube channel. In continuation with the lecture series on basics of immunology, today we'll discuss immunoglobulin E. Immunoglobulin E is also known as the reaginic antibody. Let's proceed. Immunoglobulin E was the last antibody to be discovered. So you must be wondering which is the first one. So the first antibody-like molecule which was discovered was the Benz-Jones proteins, for which I will upload another lecture. IgE is found in very low concentrations in serum, which is around 1.5 to 4.5 micrograms. Molecular weight is around 1,90,000 to 2 lakh Daltons. Half-life is very short, which is around 2 to 3 days, and serum concentration, as you know, is also very low in normal persons. Sedimentation coefficient is around 8 Swedberg units. It is a monomeric antibody having a valency of 2. Now we will see the structure of immunoglobulin E. Structure of immunoglobulin E. Let's take a look at it. So this is an immunoglobulin E molecule. Like all other antibodies, it is a Y-shaped molecule. It has 2 light chains and 2 heavy chains. In the constant region, it has CH2, CH3 and CH4. The heavy chain class is epsilon, whereas the light chain is either kappa or lambda. Immunoglobulin E is known as a cytophilic antibody. Let's try and understand why it is known as a cytophilic antibody. So this is a immunoglobulin structure having an FAB portion and FC portion. The FC portion of immunoglobulin E can attach to mast cells and basophils, and because of the attachment of these immunoglobulin E molecules to the mast cells which have receptors known as the FC receptors, these mast cells become armed to react specifically with particulate or soluble antigens present on allergens. And this is the reason why immunoglobulin E is known as a cytophilic antibody. Immunoglobulin E is also known as a reaginic antibody. It is the main anaphylactic antibody in human beings. IgE can bind with the FC portion to the mast cells and basophils by the CH4 domain, which can cause anaphylactic reactions. So reagin is basically an antibody found in the serum and skin of allergically hypersensitive people. In normal people, IgE is found in very low concentrations, or a reagin is found in very low concentrations. A reagin antibody is also an antibody that can be detected through serological tests. In the next slide, we will see the mechanism of the reaginic antibody immunoglobulin E. So let's understand the mechanism of the reaginic antibody in allergic reactions. On first exposure to allergen, the allergen is able to cross the mucosal lining. Once it crosses the mucosal lining, it comes in contact with antigen presenting cells and the T cells. These cells release certain cytokines, and these cytokines activate the B cell for maturation. On maturation, this B cell transforms into the plasma cell, which is an antibody secreting B lymphocyte, and the immunoglobulin E secreting plasma cells will release immunoglobulin E in the circulation. These immunoglobulin E molecules go and bind with their CH4 domain to the mast cells and the basophils. This is on the first exposure to allergen. Now, these mast cells bound to immunoglobulin E are present in the circulation. On second exposure to the same allergen, degranulation of the mast cell occurs in which the mast cells release various chemicals. These chemicals include histamine, kinins, leukotrienes, prostaglandins, cytokines, serine proteases. All these chemicals have an important role in allergic reactions and sometimes may also cause anaphylaxis. So, in allergic reactions can be as simple as sneezing, allergic rhinitis, skin rashes, urticaria, itching, and all the substances that are capable of causing these reactions may be some food particles or some pollen grains, dust, etc. So the person may suffer with simple rashes or itching, and it can be as severe as an anaphylactic reaction. Let us take a look at the functions of immunoglobulin E. The mast cells, on binding with immunoglobulin E, degranulate to release histamine and slow reacting substances. These chemicals may cause vasodilation, hypotension, bronchial constriction, immediate type 4 hypersensitivity reaction in sensitive people. Immunoglobulin E is also known to play a role in autoimmune disorders, and it has a very important role in eliminating parasites from our body. So, this is one area in which immunoglobulin E protects the body from parasitic infections. These are my references for the lecture. Thank you for watching. Like, comment and share. Don't forget to hit the bell icon.